So, welcome to this lecture on carbohydrates. Actually, in particular, we're going to talk about polysaccharides. So we've already talked about monosaccharides, which are individual monomers of sugar or saccharides, and now we're going to dive into when we start making these into long-chain polysaccharides. I think we've introduced the concept of disaccharides, and that's kind of in the middle, and we'll now build up these long-chain polysaccharides, and we'll also begin to address post-translational modifications of proteins, GPI anchors, and a few other things, because as we get into membranes and we get into biosignaling, those will become very important. So a quick case study, we're going to talk about Jenny Baird, who's a 34-year-old woman. She comes in for a doctor visit with influenza-like symptoms, including fever, large tender lymph nodes, throat inflammation, rash on her trunk, frequent headaches, and sores on her mouth. Personal medical history: she does have asthma. She also has acid reflux disease, gastroesophageal reflux, and she has frequent bronchitis. She's sexually active with multiple partners, history of crack cocaine use. She last used about a month ago. She smokes marijuana about on a monthly basis as an appetite enhancer. She smokes about half a pack of cigarettes per day, and she drinks alcohol at about the rate of 1 or 2 drinks on the weekends. So the question is, what does Jenny have? What tests would you run? The tests you would run, since she's sexually active, are HIV test, which in this situation was positive. They ran in the HIV situation, usually you run a quick ELISA. That's a quick test, and it's confirmed with Western blot and or real-time PCR, which you should know all of those assays at this point. They tested for hepatitis A, B, and C, all negative, mononucleosis negative, Epstein-Barr negative. Interestingly enough, mononucleosis and Epstein-Barr are both herpes viruses. So HIV is a lentivirus. It's a subgroup of the lentivirus that is a retrovirus. HIV infects T cells, CD4 plus positive T cells, macrophages, and dendritic cells, and you might note that those are all immune cells. Now, one thing that's also very important is that you might note is that there's a protein called GP120. That's one of the fusion proteins. It has a ton of glycosylation, and this will serve as the anchor point for this lecture in the grand scheme of things. So again, you might note the important part of this structure. We have DNA, we have our HIV virus with our nucleotides in the center. I'm not a HIV virologist, so I don't really recall. I think it's an RNA, and since it's a retrovirus. But the important part that we're going to talk about today is if you look at the tip of this GP120, which is a trimer on the surface, you will note that there are large sugar residues that occur, and this is called the glycan shield. And what we find is that if you take GP120 as a protein, it's anchored in the membrane. As you might recall from the previous lecture, we have proteins that are anchored in membranes. We'll actually go into a little more detail on this in the next lecture, but GP120 is anchored in the viral envelope, which is a membrane, and there are exposed residues on the surface. But what you will find that is very fascinating is if you have exposed residues, they are quickly covered up by carbohydrates, and so that's actually what the protein looks like to your immune system. It's very hard to develop antibodies against this virus, and that's why it's very difficult to generate a vaccine. It's because all of these glycans, and influenza does very similar things. Actually, all viruses do. AIDS, just HIV, happens to be one of the masters, as does influenza. There's a pymol structure of GP120, and I would encourage you, it's anchored in the envelope. I would encourage you, it's at the end of this exercise, I'd encourage you to look at it and pop out some of these residues that have glycosylation sites on them, and from there you can really see what's going on with this disease. It's really fascinating, actually. It's one of the reasons why I really enjoy pymol, is when you put all those in there and you show them and show how many glycosylation sites there are, it really kind of brings into focus an understanding of why it's difficult to develop an antibody. It's also a molecule a month, that's why this picture's in there, and I would encourage you to look at that because it's a very interesting story there as well. Again, this is the crystal structure with carbohydrates, and you can just see how this is the trimer. This is looking at it from the top of the, and you can just see how much carbohydrates obscure the protein epitopes. Carbohydrates are generally not very immunogenic, so they don't generally raise a whole bunch of immune systems, immune response. Although they can, they generally don't, and this is why obscuring all the epitopes, all the protein epitopes with these glycans, these carbohydrates, these sugars, kind of is a really cool phenomenon. Again, so we'll come back to this picture, but remember how this looks, and you'll understand what we're talking about. Again, GP120 is a trimer, and they have all these structures that exist, these carbohydrate structures that exist on this protein. Okay, so let's go back. Let's dive back in. So we started with disaccharides last time, and we'll dive back in there, and then we'll just keep going with the oligosaccharides. So two sugar molecules, two sugar monomers can be joined together by a glycosidic bond between the anomeric carbon and the hydroxyl carbon. And this glycosidic bond, which is an acetyl between monomers, is less reactive than the hemiacetyl at the second monomer. And the second monomer with a hemiacetyl is reducing. And so what we end up with, an anomeric carbon is generally is involved in the glycosidic linkage and is non-reducing. So again, lactose on the beta-1 anomeric carbon links up with the 4, the oxygen, the hydroxyl on the 4 of a glucose, and you end up with the beta-1,4. In sucrose, we have a alpha 1, 2, alpha 1, beta 2 glycosidic linkage. In trehalose, we end up with an alpha 1 to alpha 1 glucose to glucose linkage. So different, both sucrose and trehalose again are non-reducing. So the non-reducing end, which is the anomeric carbon end. Sucrose, again, you can see this gives us just a little bit more insight. Sucrose in there, we have our, gives a little bit more of a graphic representation of what's going on. And sucrose, again, is between our, sucrose is glucose and fructose. And so you can see the fructose, both are anomeric carbons, and therefore there's no reducing end on this. The reducing end would be the anomeric carbon, so sucrose is a non-reducing disaccharide. Lactose, it's principal sugar present in milk. It's in about 5 to 8% in human milk and 4 to 5% in cow's milk, so it's a significant portion. There's a beta-1,4 glycosidic bond between a galactopyranose, 2-galactopyranose. It's a, again, beta-1,4 glycosidic bond. Maltose, some of you are probably pretty familiar with maltose as malt. It's from malt, I guess I should say. It's a juice of sprouted barley and other cereal grains. You have an alpha-1,4 glycosidic linkage there. Again, the anomeric carbon is linked up to the hydroxyl, which is the 4 carbon, and you have 2 glucoses in this situation that are linked together. Now, you can have, besides a disaccharide, if you keep adding on to the reducing end, you can keep building up chains, and you can end up with homopolysaccharides. If they're then they can be branched as well. You can also go off the 6, so generally go off the 4. We can also branch off the 6 on the same molecule, the 6 carbon, which is the carbon that is at the terminal end of the molecule. It has a hydroxyl group in there that you can branch off of, and so you can have a homopolysaccharide where all of the sugar units are the same, and we can branch off of a 6 and create an entirely new rung, so to speak, an entirely new branch of put together glucose molecules or sugar carbohydrate residues. You can also alternate between 2 different monomer types. You can have multiple monomer types. You can branch multiple types, and so there's a whole complex of ways of doing this. Again, a polysaccharide is a carbohydrate consisting of a large number of monosaccharide units joined by glycosidic bonds. And starch is an example. Amylose is an example. We'll talk. Amylopectin is an example. And these are all great sources of energy, and we'll talk about all of them as we get going. Glycogen is one of the main forms of energy. It's a branched homopolysaccharide of glucose. Glucose monomers that are formed together in alpha 1,4 glycosidic linkages. There are branch points within this, alpha 1,4,1,6 linkers, about every 8 to 12 residues. That linkage, that branch point, provides disease states in certain people who do not have an enzyme to cut off that branch. We'll talk about that a little later. The molecular weight of this can reach several millions, and its function is the main storage polysaccharide in animals. Glycogen does. Starch is a mixture of two homopolysaccharides of glucose. We have amylose and amylopectin. Amylose is an unbranched polymer alpha 1,4 linkage. Amylopectin is branched like glycogen, but the branch occurs about every 1 to 24. Occurs to every 24 to 30 residues. The molecular weight of amylopectin can be up in the 200 millions. Starch is a main polysaccharide in plants. And here we kind of see how this works. So we have amylose, which is glucose units, alpha 1,4 linkages. We have a non-reducing end on one end, that's the terminal end, and then we can just keep adding on to the reducing end. And we can also branch, as you can see here, and instead of going linking on, we can link on and the alpha 1,6 on the branch, off the branch, and form new branches. So we can also, so this one on the main chain, there's a link off the 4 and the 6 carbon, both hydroxyl groups. So that's very common. Again, you can also have mixtures of these, amylose and amylopectin and starch, and they'll form double helices just like protein, just like DNA does. And this is actually a very interesting structure. There's several structures in the NIH 3D printing library if you have an interest in looking and seeing what this looks like and how it's how the scale and relationship to DNA. Again, polysaccharides, we can have 1,4 linkages and we can have alpha 1,6 linkages. These are, again, provide I would know this inside and out, these polysaccharide linkages and the types. I would be able to identify an alpha 1,6, an alpha 1,4, a beta 1,4, and understand exactly what that means. Metabolism of glycogen and starch, and we'll go into great detail on this, but just as an introduction to next week's topic, glycogen and starch often form granules in cells. These granules contain enzymes that synthesize and can degrade these polymers. Glycogen and amylopectin have one reducing end, but many non-reducing ends. So enzymatic processes occur simultaneously in many non-reducing ends. So once it goes in, it's going. So humans and animals cannot digest cellulose, which is the main plant carbohydrate, like trees, because we don't have beta glycosidases. That's one of the reasons why we can't eat trees. But termites and other bacteria do, and some of the ruminants have bacteria, such as horses and cows have bacteria that can break down that bond. I think it'd be very interesting. I'm not quite certain why we haven't done this to build. Humans only have alpha glucosidases. It'd be kind of interesting to give us a beta glucosidase, seeing if we could actually eat trees. That's just biotech, and I don't know if it's realistic or not. Or we could just give ourselves microbiome update that have bacteria that can eat those. And I don't know the answer to why we don't do that. It's just an interesting idea. I'm sure someone's thought of the idea. One of the other important things that you'll see in trees and plants is the hydrogen bonding that exists between carbohydrates, and this is obviously, this structure right here kind of shows that nature. So when you start getting these structures, that's one of the reasons why trees are so, and that you have extreme rigidity in these molecules, and water is also an important part of this. So this hydrogen bonding plays a critical part in the strength of many plant structures. Okay, so that's polysaccharides, and that's the summation of, I guess the important thing is you understand the glycosidic bond inside and out and be able to identify it. So let's talk about some of the other carbohydrate that exist. Some of the other important carbohydrates. There's glycoconjugates, where we're going to put sugar and you're going to interact with other biopolymers. So the first one, and probably the most prevalent, is glycoproteins. These are proteins with small oligosaccharides attached to them. reference back viral proteins, GP120, that has huge glycosylation off that protein. About 500,000 proteins are glycoproteins, and they're basically carbohydrates that are attached covalently to certain residues inside or on a protein. Carbohydrates play a role in protein recognition. It's interesting to note that bacteria does not post-translationally modify proteins, and so they do not have a lot of glycoproteins. There's very few that have it. Viral proteins, on the other hand, are very heavily glycosylated, and this helps them evade the immune system, if you reference our case. So again, glycoprotein is a protein with a small oligosaccharide attached. The carbohydrate is attached via its anomeric carbon. About 500,000 proteins are glycoproteins. Again, carbohydrates play a role in protein recognition, which we'll go into some detail in this lecture and in some other detail in the biosignaling lecture. Only some bacteria have glycosylation, so not very many of them have glycoproteins. On the other hand, viruses are heavily glycosylated. That helps them evade the immune system. In fact, almost all viral fusion proteins are heavily glycosylated. Oligosaccharide linkages in glycoproteins. This, so serine and threonine, as well as a few others, as I'll show you in the chart, provide the opportunity to be O-linked or N-linked. And so an O-linked attaches to the hydroxyl group on the anomeric carbon. That's, for example, serine and threonine, and you can add in whatever flavor of the month carbohydrate you'd like to add. Asparagine also you can add on the amine group, on the hydroxyl group, in so that we get a amine linkage there on asparagine. That provides the linkage there on N-linked glycosylation. So O-link stands for oxygen, i.e. serine threonine. Asparagine is N-linked because it's nitrogen-based. So that provides you an understanding of what that general principle is. Again, O-glycosylation, serine, threonine, tyrosine. Some of the functions of that include extracellular matrix formation. It helps with the protein folding, protein localization, and many others. And basically, you can get these large. So this graphic shows a protein that's folded in a membrane, and you can add these large complex structures where you have glucanac, mannose, and then a branch, mannose, glucanac again, galactose, followed by a sialic acid. All of these are carbohydrates. And glycosylation, there are multiple enzymes that put this together. Again, same thing that can happen. Again, it's with lysine and asparagine, which obviously have amine functional groups on the end. It can be, again, for protein structure, protein folding. It can localize your protein to in a specific cellular compartment. And it's involved in numerous other post-translational modifications. As you might note, these are covalently linked modifications. I will introduce this concept now, and we'll talk more about it when we talk about membranes and when we talk about biosignaling. We can add, we've already introduced the concept generally, but we can add different functional groups to proteins. If you look at this chart, O-glycosylation and glycosylation are the examples, and you can see down at the bottom of the post-translational modification the residues that are able to be modified in this, and there's a few others in this chart. So this kind of gives an idea. So if we have a protein where we have serine, glycine, X stands for any amino acid. Glycine is the core protein. If this is the N terminal, this is the C term. We'll take the serine. We can put together a long carbohydrate chain, and we put together, again, linkages, glycosidic linkages together, and we can look up and put chondroitin sulfate on the end of that and link those together. And the reducing end is down at the end here. All of this is attached to the serine covalently. We also have two family members of membrane proteoglycans where the protein is anchored. It has there's a transmembrane domain or a GPI anchor. GPI is a phosphatidylinositol anchor in here. So we got a lipid that from yesterday's lecture that anchors in the membrane, or you can have a transmembrane, very hydrophobic residues that cross the membrane, and then we end up with these sulfate or another chondroitin sulfate. You can end up with these carbohydrates that link off this protein. And so the other thing that we have is we have so this is a syndecan where the protein has a transmembrane. Glypican has a GPI anchor, and you notice we have a carbohydrate residue through here, and then we have a protein that is that has heparin sulfate, which is a carbohydrate. Linkage is off there. So all of these provide important interactions. And they have important functional properties. They can also get clipped to the GPI anchor if they need to. Glypicans have 14 conserved cysteine residues that form these disulfide bonds that allow the molecule to kind of form in a globular nature. Again, sulfated glucosaminoglycans attached to large rod-shaped proteins in cell membrane. That's a proteoglycan. We have syndiclans and glypicans. Again, they're anchored in and they interact as receptors for neighboring cells, and they regulate cell growth. So basically, they're cell-to-cell receptors. Another important linear polysaccharide found in animal tissues is heparin. It's a proteoglycan in which 2 or 3 heparin sulfate chains are attached in close proximity to a cell surface or an extracellular matrix proteins, and it regulates important developmental processes, including blood coagulation, angiogenesis. You're probably pretty familiar with heparin and heparin sulfate. Heparin sulfate is a very important cellular receptor for viruses. Herpes virus. Numerous herpes viruses bind to heparin sulfate. These heparin sulfate can be quite large, and they form these extracellular matrix compounds or complexes that have all kinds of growth factors and extracellular enzymes, coagulation factors, cytokines, chemokines that are associated with them. Again, heparin is a repeating pentasaccharide unit of heparin. Heparin has a lot of sulfate in it that's very charged. It's very negatively charged, and that functional charge creates a very defined structure-function relationship. Again, this kind of gives you an idea, so we can attach these heparin sulfate chains, and this gives you an idea of what heparin sulfate is. So we have a glucanac, a glucose, a modified glucose, aminoglucose, and an amine with a sulfur on it, glucose, and we also have other sulfated carbohydrates, and we end up with these large structures that help build up this heparin sulfate chain. And this is kind of the general structure where we have NS2S, NS2S, NS2S. So that's the domain of that heparin sulfate in proteins. Again, proteoglycans have different glycosaminoglycans, which are carbohydrates with amino groups on them that are linked to the core protein. They link from the anomeric carbon of xylose to the serine hydroxyl group. We have many tissues have different core proteins, and aggrecan is the best studied. We also have glycolipids. A lipid is a covalently bonded oligosaccharide. Parts of plants and animal cell membranes have this lipid that inserts into the membrane with glycans, with carbohydrates that stick out. And this glycan, as you might recall from yesterday's lecture, we have ganglioside carbohydrate, which determines our blood group, and we also have a very also important glycolipid, which is LPS, lipopolysaccharide, which covers the outside of a bacteria in some bacteria and defines it's the primary defining feature in gram-negative versus gram-positive bacteria. Here's a picture of that lipopolysaccharide in the outer membrane of salmonella. Again, 6 fatty acid residues in the lipid portion of the molecule, and that's a little bit different. That varies a little bit per bacteria. They have that common lipid region. Then we have a core polysaccharide region, and then we have an O-specific chain of additional carbohydrates. So we also can get proteoglycan aggregates, hyaluronan, HA, and aggrecan. Again, as I mentioned a little bit earlier, form huge non-covalent aggregates. They can hold lots of water. They make great hydrogels. They are low friction, and they cover our joints, our articular cartilage, by reducing friction, and they also provide load balancing. So they're 2 times 10 to the 8th. They can have a molecular weight up to that size. So this is a picture of hyaluronan, and you have all these keratin sulfate with their linked proteins of chondroitin sulfate. So keratin sulfate and chondroitin sulfate are basically the feather parts with the protein as the backbone. So hyaluronan can have up to 50,000 repeating disaccharide units, and we also can have aggrecan as the core protein that's associated with these molecules. They're very, very large. And a lot of them have a ton of non-covalent interactions. So, and that non-covalent interactions allows for, again, significant hydrogen bonding to occur, and it allows water to be, it allows this to be a hydrogel where it can retain water. So let's talk a little about extracellular matrix. Extracellular matrix is the outside of the cell, the strength, elasticity, and the physical barrier in tissues. Its main components include proteoglycan aggregates, as you just saw, HA, and aggrecan. Collagen is also, and elastin are also in there. So it's a barrier for tumor cells. It's a barrier for other bacterial invaders. Some tumor cells secrete heparinase that degrades the extracellular matrix to allow it to invade. So sometimes we attack our cells with cancer. Cells interact with this extracellular matrix. Some of the membrane proteins that are important proteoglycans include the syndicans. Other integral membrane proteins are receptors, integrins. We'll talk about integrins when we talk about biosignaling tomorrow. These proteins link cellular cytoskeletons to the extracellular matrix and transmit the signals to regulate important body things like cell growth, cell mobility, apoptosis, wound healing, all kinds of stuff. So these proteins link out into the extracellular matrix, and there's a signaling molecule on the inside of the molecule that allows the molecule to respond to different conditions, as you can see from here. So that's how a cell interacts with the extracellular matrix in the outside world. And this is kind of what it looks like. And again, we'll go over this in a few days. We have an integrin or tomorrow. We have an integrin that goes through a extracellular matrix into an actin filament. That actin filament, and we can have integrins integrate in with all of these cross-linked collagen and proteoglycans and fibronectin, and that kind of anchors that cell inside a tissue. So that provides the extracellular matrix, provides the strength, elasticity, protection from immune system, and all that fun stuff all goes on. And these are large carbohydrate structures, large proteoglycan structures with proteins and carbohydrates intermingled in there, as well as membranes. And the membranes provide the lipids. They provide the important structural component as well. One other last thing that's kind of important, I shouldn't say last thing, but one other thing that's very, very important with oligosaccharides is in cell recognition. So viruses, bacteria, even our own immune system recognize different presentations of oligosaccharide chains in order to provide, and also enzymes provide important abilities and important functionalities. Obviously, viruses use oligosaccharide chains like heparin sulfate to bind. We can also have toxins that bind to extracellular matrix carbohydrate chains, as well as bacteria. P-selectin binds leukocyte extracellular carbohydrates. Enzymes also have mannose-6 phosphate receptors like lectins on them as well. And so all of these things, oligosaccharides, are very important in cellular recognition. Again, so the nature of pathogenic agents binding to carbohydrate ligands on host cell surfaces, bacteria, viruses, and toxins can overcome these low affinity binding by combining interaction sites. So they commonly you'll see these lectins and other things that will add up low molecular interactions, low strength interactions to build up, and then allow them to attack. So adhesins, which we'll see in the next lectures, are presented, and these viruses, you have microbes, you have toxins that all can attach. This just gives another repeat of the 4 different types. So adhesin analogs are available, microbes are able to attach to. Receptor analogs, large soluble conjugates are able to attach. And antibodies against glycan lectin envelopes are also able to attach and prevent. So all these things can occur. So we can have large-scale soluble conjugates that occur, and all of these things provide interactions in a cell. We also can use these to make nanoparticles, and dendrimers, which is something I've studied quite a bit of, provide an excellent example of nanoparticles of carbohydrates. These are used in hydrogels, which are important in things like microarrays and other surfaces. These hydrogels allow a water interface, a water shell, to be maintained without evaporation, because evaporation will occur very, very quickly. So these, so we have in these nanoparticles, we also have protein. We have different mixtures of proteins, carbohydrates, and inorganic molecules and emulsions that allow for different properties and different functional opportunities. Okay, so I told you at the beginning of the lecture I was going to re-show this. This is GP120 on HIV, which is the fusion protein for HIV. And you can see what GP120 does is put these little circles and squares in different colors represent different carbohydrates, different monomers of carbohydrates, and GP120 is building up this glycan shield, building up these carbohydrate structures so that the immune system cannot recognize the virus. And that's one of the main reasons why HIV is so difficult to develop a virus against and so difficult to fight off. Thank you for listening. We're excited to finish and wrap this up on carbohydrates. I think, again, the important points have been very, very well emphasized. And I encourage you to look at this pymol activity and see where the glycosylation sites, just color them. If you'd like to have me do this for you, I'm happy to do that. I think this is a very, very interesting and fascinating subject matter, and I have a collaborator at the University of Pennsylvania who studies this phenomenon in influenza, and it's very, very fascinating how viruses use this phenomenon, use glycosylation in order to evade the immune system. Look forward to discussing this topic with you.